Right, I'm going to start off with some basics. So you should have the title Protein Synthesis, the Basics in your contents page and in your next page in your notebook. Okay, so what I'm going to do is I'm going to give you a really a big overview, and I got to throw in one or two little bits of review as well. So let's start off here with this is a strand of DNA. How do I know this is DNA? Well, it's got certain clues about it. First, I know it's DNA because it's a double helix. Okay? Second, I know it's DNA because it's made from nucleotides that contain the sugar deoxyribose. Okay? You notice it ends OSE. That means it's a sugar. And the deoxyribose, that's the D in DNA. The other thing that I know that it's a DNA is this. The nitrogen bases which make up the nucleotides are either A, T, G, or C. And finally, if there is an A on one side of the DNA strand, there's going to be a T on the other. And if there is a G on one side, there is going to be a C on the other. We have what we call complementary base pairing. A always goes with G and G always goes with C. Okay, that's DNA. The other thing is, if I'm going to go on a hunt in the cell for DNA, there is only one place that you ever find DNA. Only one place you find it, in the nucleus. You will never find DNA outside of the nucleus. Okay, that's DNA. Now, there is another type of nucleic acid. We haven't really talked much about it yet because we haven't had to. And that nucleic acid is called RNA. Okay, RNA. And, okay, so here, how do I know if I'm looking at DNA or RNA? Well, the first thing is, you'll notice with an RNA, it is a single helix. It does not have two strands. Okay. The next thing is, if the D in DNA is for deoxyribose, the R in RNA is just for ribose. So when you look at the nucleotide, remember nucleotides, phosphate, the sugar, and the nitrogen base. The sugar part, this part here, in DNA that's deoxyribose, in RNA it's just ribose. That's the R in RNA. Now, another thing that is a little bit the same and a little bit different is the nitrogen bases in DNA, in RNA. Okay, we still have A, we still have G, we still have C. So we have adenine, guanine, and cytosine. But instead of thymine, we have the nitrogen base U for uracil. So we have A, G, C, and U. And the U, this nitrogen base here, is uracil. So not only it doesn't end INE like all the other ones do, it's called uracil. Okay, so if ever you see a nucleotide with U, or you see a nucleic acid with U, it's going to be RNA. It can't be DNA because DNA does not have U. DNA has T, and RNA does not have T. RNA has U. Okay, now, complementary base pairing. Well, since it's only a single strand, in the actual strand of RNA, you don't have complementary base pairing. But if this ever base pairs up with DNA, which you'll see, instead of A going with T in RNA, A goes with U. Okay, another thing that's slightly different about RNA is this. Whereas DNA is DNA is DNA. There's only one type of DNA. RNA can be three types. We have mRNA, where the M stands for messenger. Okay, we have tRNA, where the T stands for transfer. Okay, and those are really the only two that we need to really know in detail about. And then we have rRNA, right, where the R stands for ribosomal. Now you're thinking, ribosomal sounds a bit like ribosome. Well, you would be correct, because ribosomal RNA is what makes up ribosomes. Okay, messenger RNA, transfer RNA. They're the same basic single strand, but they're split with these letters. All right, this isn't just RNA; it's messenger RNA because it's a messenger, and this isn't This isn't just RNA; it's tRNA because T stands for transfer, which is part of its job. So these little letters here tell you kind of what job it's doing. Okay, that is kind of the basic difference between DNA and RNA. DNA, double helix, deoxyribose sugar, nitrogen bases, adenine, thymine, guanine, cytosine, only found in the nucleus. Messenger RNA, found throughout in different places in the cell, including the nucleus. It's a single helix. The sugar in the nucleotide is ribose. Let's call that nucleotide. Okay, building block of nucleic acids, and its nitrogen bases are A, G, C, and U for uracil. Okay, now what we need to know is this: how do we start with a double helix strand of DNA and end up with a protein? Because proteins are a different biomolecule to DNA. DNA is a nucleic acid, proteins are proteins. And that's the process bit where we're talking about the process and the purpose of protein synthesis. Step number one, and I'm going to make this really simple because the next video will explain this in a little bit more detail. Transcription. Step number two is translation. How do I remember which one's which? If somebody comes up with a good way to remember that transcription comes before translation, let me know, because all I ever use is that is alphabetically before translation. Okay, here's what transcription is. We start with a double strand of DNA and we use it to make a single strand of messenger RNA. That is it. That is transcription. Nothing more than that. We're using our DNA as a template to make messenger RNA. Right? This, because we're using DNA, happens in the nucleus. Now, the messenger RNA then, because it's a messenger, it leaves the nucleus and goes to a ribosome. Okay? So this is nucleus. This is going to happen in a ribosome. Okay? And a quick reminder, where are the ribosomes? All these little black dots on the endoplasmic reticulum are ribosomes. You will notice now they are bang smack right next to the nucleus. They are found bang smack next to the nucleus for a good reason, because the closer they are to the nucleus, the less distance that messenger RNA that we've just made has to travel. Makes perfect sense to me. Okay, so in translation, we are going to take a strand of messenger RNA, okay, with its nitrogen bases on it, and we are going to use it to put together amino acids, right? Now, amino acids, if you recall, are the building blocks of proteins. So if we put these amino acids, each one of these is a different amino acid, and they're joined together. They have to be joined together in exactly the right order, because when amino acids are joined together in exactly the right order, you make a protein that has exactly the right shape. And if you remember nothing more from enzymes, which I'm hoping you do, you will remember the active site of an enzyme, which is a protein, has to be a perfect fit for its substrate or it can't do its job. That is true of all proteins. All proteins have to be exactly the right shape to be able to do their job. So when we put the amino acids, right, we bring them together in exactly the right order. They join together. And what do you call amino acids which are all joined together? You call it a protein. And so translation is using the instructions on the mRNA, the messenger RNA, to put amino acids together in the right order. And that is the basics of protein synthesis. Next, I'm going to show you this in a diagram form.